In eukaryotes, the 26S proteasome is responsible for regulating the concentration of hundreds of regulatory proteins and degrading damaged and misfolded proteins. The proteasome is also involved in multiple important cellular processes like cell signaling, DNA repair, and mitosis. For a protein to be targeted to the proteasome for degradation, it must meet two requirements. The first requirement is a ubiquitin tag, represented here by the chain of blue circles. The ubiquitin tag targets the protein to the proteasome by binding to ubiquitin receptors on the proteasome. The second requirement is an unstructured region, represented here by the red line. The unstructured region allows the proteasome to grab onto the protein and begin degradation. My project is to make various model proteins that have both requirements of a ubiquitin tag and an unstructured region and test if they're degraded by the proteasome. Each of my model substrates also have a green fluorescent domain, represented by the green square, in order to track the degradation of my proteins. I express my proteins in E. coli cells because bacteria grows rapidly and has the ability to make tons of protein in a few hours. The E. coli cells make a bright green protein, like this, due to the green fluorescent domain in my proteins. Next, I purified my proteins of interest from the E. coli cells, and we can do that using a protein purification column like this one. My model proteins have a specific tag that binds to the beads in the column, trapping my model protein in the column. All other unwanted proteins do not have this tag, which allows them to flow through the column. Finally, when I incubate my model proteins with proteasome, I can measure degradation by tracking the green fluorescence of my proteins. As the time increases, the proteins will degrade and disappear, which means that the fluorescence detected will decrease. From this, I can plot the fluorescence over time and make a graph that looks like this to track the degradation rates of my proteins. So why is this important? There are some neurodegenerative diseases that are characterized by the buildup of protein aggregates. Theoretically, these proteins should be degraded by the proteasome, but they're not. My project will allow me to better understand the mechanism of protein degradation by the proteasome that can hopefully be applied to treating neurodegenerative diseases in the future.